Here we are again with a new chapter, that is cell cycle, cell division and chromosomes, all about cell, right? So, cell division is one of the really important thing that happens within a cell. With the help of cell division, a cell divides into two, and that is how life is going on. That is how we grow. We imagine yourself in nursery, imagine yourself before nursery, and look at you now. All of you have grown up, right? This is all because cells are dividing, right? So cell division is playing a major role in the growth of organism, in reproduction part, and in repairing of the cell when there is some kind of injury and new cells are needed, then also cell division is taking place. Or say when you are having some kind of that the old cells are continuously being shut off, they are dying, and in order to replace the old cells, then the new cells are taking their place, right? So this is all happening just because of the cell division, because cells are dividing, right? So cell divides, right? We know this. So how? What is it inside it that is splitting itself? So let us know about the chromosomes. We know cell has a nucleus. Inside the nucleus is present DNA, the nucleic material, right? So this DNA is organized into some units, and such units are called chromosomes. Okay, chromosomes is having two parts. Chromosomes are having two parts: DNA and proteins. DNA is always present along with the proteins, and this part where DNA, proteins, they lie together, they together are known as chromatin. Okay, DNA is never alone, it always works with the proteins, and this association of DNA plus proteins is known as chromatin. The chromosomes very firstly were discovered by Walter Fleming. He was studying about a salamander. Salamander is an amphibian. At that time, way back, the microscopes were not so good, but now because there is much of the invention in case of the microscopes, so we can see the chromosomes really in a better way, right? So let us continue with that. So okay, chromosomes are not seen if the living cell that you're placing under a microscope is not stained. You have to put some dye in order to see the chromosomes. So the chromosome name itself means coloring body, because they adapt to the color, whichever dye you're using, pink, blue, green, whatsoever. So they'll pick that color up and you'll see them under the microscope as those colored bodies, right? Moving forward, chromatin is made up of two things: DNA and histone. So DNA, the very first structure of DNA was given by Franklin. But after that, the model which we study nowadays is given by Watson and Crick. So Watson-Crick model of DNA, how does it look like? They say that DNA is a macromolecule, quite a big molecule, and it has a double helical structure. Now this structure is really important, they can ask you. Double helical structure. They say that DNA is having two strands, two threads, two strands which are wrapped on each other. Okay, and each DNA is having repeating units of nucleotides. On DNA itself are present the nucleotides. Now nucleotides are made up of three components: sugar, phosphate group, and a base. So the sugar and phosphate group, they remain same, but the bases keep on changing. And there are four bases. Number one is adenine with A, guanine with G, cytosine with C, and thymine, okay, with T. And remember this, that adenine always combines with the T. and C always combines with the G. When A and T combine, they always make a double bond in between them, and when C and G combine, they always form a triple bond. Okay, and these are the hydrogen bonds made up of hydrogen, right? 4 bases, adenine, guanine, cytosine, thymine. A always combines with T and C always combines with G. Alright, so on the DNA, these nucleotides are present, right? But considering the fact that we do not write all of this part, we don't draw sugar and phosphate, but we just write the base A, T, C, G, A, C. Okay, if these are present on one strand of DNA, the other strand will always have the complementary base pair, which means if A is present here, right opposite to it will always be present T. If T is present on this side, then here it will be A. If here is C, it will be G, then C, T, G. Alright, so this is how the strands work. If one strand is having one molecule like this, the another one will have the complementary base. This is why the another strand of DNA is known as complementary strand. Okay, so this is how DNA look like. This is the Watson and Crick model. This is known as double helical structure of DNA. Okay, so this DNA is always wrapped on histones. So there are always 8 protein molecules. These proteins are special proteins known as histones. So 4 ahead, 4 beyond. And DNA will take 2.5 rounds on 8 histones, then move further, then another 8 histones. I am drawing 4 because 4 are just behind it. Then again 2.5 rounds, then again 4 histones, and like this. So DNA moves on like this. DNA is not present all alone. It is always present along with the histones, and this DNA and histone, this one part, this is known as nucleosome. 2.5 rounds of DNA with 8 histones forms one nucleosome, right? And all the nucleosomes together form a chromatin. Okay, chromatin consists of DNA and histone. DNA is always wrapped on histones, but this one molecule which is being formed, which is a mixture of 2.5 turns of DNA and 8 histones, this forms one nucleosome, and there are millions of nucleosomes present inside one single nucleus. Okay, so inside one nucleus, DNA of approximately 2 meter is present, and while the size of nucleus, diameter of nucleus is just 6 micrometer. So this 2 meter DNA has to adjust inside the 6 micrometer nucleus, which means the DNA needs to be highly coiled. Like telephone wire, you can see coiled. Telephone wire is nothing. DNA is really highly coiled, super coiled structure. It is forming itself in such a way, this is coiling. This turns, this everything is done for the coiling part. So it has to adjust itself within the 6 micrometer sized nucleus. It's a tedious job, right? Talking about structure of chromosome. So chromosome, as we said, is the condensed material. You know, when cell hasn't started dividing, we see a highly structure like this, which is known as chromatin. But when the cell starts dividing, it takes the shape of a chromosome. Number of chromosomes are present there. Chromosomes, these are 2 sister chromatids. They are known as sister chromatids, and these 2 sister chromatids are attached with one another at a point known as centromere. Okay. Next part is genes. So what are genes? We continuously say that the parents' genes has been transferred to the offspring. You are having your parents' genes. Your hair is straight just like your mother. Your nose is just like your mother, and etc. This part is like your father. So we say the genes are being transferred from one parent to another. Oh, sorry, one generation to another, right? So genes, what are they? Genes are, we just said this is what a DNA look like. So DNA was having complementary base pairs on it. This sequencing of 3 nucleotides. 3 nucleotides together forms a sequence for one gene. Okay, we all are having different genes because this DNA sequencing is never same, right? It's never same. Only 4 pairs are there which are present. Still imagine how much diverse the population is. Not even the 2 twins match exactly, right? So this is how the nature has decided to adjust the base pairing. Okay, so talking about the sequencing, 3 nucleotides together form a gene, right? Now if we say, imagine that ATT, let us say, is a gene for straight hair, right? If another person is having gene like TAT, this might be for curly hairs, curly hair, not hairs, right? Another one can be TTA. This means that this person is having neither too straight nor too curly, but a mediocre kind of hair, right? So this 3, the pair or the sequence of 3 nucleotide forms a gene, and if this sequence is present, then that person will have straight hair. If this is present, then that person will have curly hair, and let us assume that if this is present, that person will be a mixture of both, somewhat curly, somewhat straight, right? Average kind of. So gene, how is it transferred? So whenever the parents, they form gametes, while the formation of gametes, we'll study this ahead as well, they only give their one strand of DNA. Okay, one gamete has one strand of DNA. This, if this is father, this is mother, one strand of DNA will be from father, one will be from mother. Then both these strands, when gametes will fuse, zygote will be formed. At that time both the strands will meet, and hence you are having some of the characters of your father, some of your mother, because the genes are getting mixed up. And you will also be different from both because there will be mixing of the base pairs, right? Some of the sequence will change as well. This we'll study further. Before that, why do we need new cells? This I already told you, for growth, for replacement, for repair, and most importantly for reproduction part. Now talking about cell cycle. So cell cycle, okay, they say that in order for a cell to divide, it must complete some tasks. Okay, if a cell wants to divide, it needs to do some things, some tasks. Okay, and such series of tasks, such series of events that a cell has to go through before the division, that is known as a cell cycle. Cell cycle has 2 phases. One is interphase. This is supposedly said that interphase is something in which cell is resting, but on the contrary, the cell does not at all rest in this stage. On another hand, there is another phase known as division phase or the dividing phase. So firstly, the cell prepares itself for division in the interphase, and then it starts dividing in the dividing phase. What is happening in interphase? We'll draw a small cell cycle. Imagine this is a cycle, not that cycle, but that cycle. You know what a cycle is. Okay, there is one phase. We are talking about the parts of interphase. Interphase has 3 phases: growth 1, synthesis, growth 2. This bigger one is growth 1 phase. then there is small synthesis phase, and then there is growth 2 phase. So in growth 1 phase, this growth 1 phase, part of the interphase that we are talking about, in this RNA and proteins will be synthesized. RNA is always single stranded, its full form is ribonucleic acid, while DNA is double stranded and its full form is deoxyribonucleic acid, right? So some of the proteins and RNA which are further needed during the division is formed in growth 1 phase. After the growth 1 phase, cell has 2 options. It can either enter into the synthesis phase or it can return back to the resting phase. If it doesn't want to divide, it will go for resting, and if it wants to continue, then it will enter in the synthesis phase. In the synthesis phase, more DNA is synthesized, and remember, in the synthesis phase, the DNA is duplicated, it gets doubled. And in growth 1 phase, cytoplasm, the volume of cytoplasm, the amount of cytoplasm also increases, because when one cell has to divide into 2, it obviously needs more of the cytoplasm. In synthesis phase, the DNA gets doubled. And in growth phase, second growth phase, if some of the proteins are left to be synthesized, or if some of the other products are needed, then they are synthesized as the growth 2 phase. After that, the cell actually enters into a dividing phase. We'll talk about this dividing phase later. Firstly, we'll continue with this. We said that this DNA gets doubled up. So what happens? We said DNA is having this double helical structure. So the DNA starts unzipping itself. It gets opened up, and we said RNA is being formed. Now RNA is single stranded, right? When DNA opens up, then RNA, with having the complementary bases, starts joining this single strand of DNA. Another RNA will come and combine with this strand of DNA, and at the final, 2 separate new DNAs will be created, right? And this is known as the duplication of DNA. DNA is creating its own copy. So this process is known as replication. The word replica means copy. This is not given in your book, however, but DNA creates another DNA just like it. So this process is known as replication. Okay, so in synthesis phase, DNA is being doubled. Okay, DNA is creating a copy of its own. Now, if we are talking about the cell cycle thing, do you think that the cell cycle continues? No. In some of the cells, this cycle doesn't even work. Like the cells of your brain, the nerve cells, they are formed only once. After that, they do not divide ever. So their cell cycle is completely, permanently stopped. There are some cells like the cells of liver which has to divide like once a year. So the cell rests for a year, and after that it starts dividing. So this we call as a temporary cell cycle gets stops temporarily, right? But if the cells starts growing or dividing continuously and it cannot be stopped, then in that case we say that it is cancerous. If the cells, they continue with their cell cycle, right, even if they are not needed, if the body is not demanding of that cell division, if the brain is not demanding of the cell division, even then if the cell is dividing, then we call that cell as a cancerous cell, and then tumors are formed. And as you know, tumors are sometimes they can be, most of the times they are life-taking, right? So now in another second video, we'll talk about the cell division, the types of cell division and how this part works.